Is the slide visible?
Yes, ma'am, it is visible.
Okay. So, good evening. Hi. I'll be
Good
taking
evening.
the NPTEL live session week 3. My voice is clear, right?
Yes, ma'am, your
Okay.
voice is clear.
So, we are here for the course Cell Biology, Cellular Organization, Division, and Cell Processes. So, this is the week 3, which includes the syllabus of S phase, DNA replication, part 1, 2, and 3.
Okay.
So, starting from the questions which are present in your NPTEL assignments. First question: ubiquitin is attached to which position of the lysine amino acid side chain? So, is it alpha, beta, epsilon, or delta?
Ma'am, it is, I think, I'm silent.
Epsilon, right? It's epsilon. Okay. Question number 2: which of the following factors is not required for formation of pre-replicative complex on origins? ORC, origin recognition complex, CDC6, phosphorylated MCM proteins, or CTT1?
Ma'am, I think it is phosphorylated MCM protein.
Okay, right, correct, good. Coming to question number 3, which of the following is present in proteins of the ubiquitin-like protein family? Is it 5-stranded beta sheet, alpha helix, short 310 helix, or all of the above?
Ma'am, beta helix?
Beta sheet,
Yes, no, ma'am, sorry, alpha helix, sorry.
alpha helix, okay. It is all of the above. I will explain later. So, what do you know about ubiquitin-like protein family?
Ma'am, actually, I want to know that it is third week ना. Actually, मैंने बहुत लेट में enroll किया था.
Okay.
तो मैंने फर्स्ट वीक और सेकंड वीक नहीं किया था, बट एज यू नो दैट कि 70 आई थिंक 70% और 80% असाइनमेंट इज कंपल्सरी आई थिंक.
Yes, yes.
तो मैम मुझे प्लीज आप एक वैसे डाउट सेक्शन है तो मुझे सेशन है तो मुझे प्लीज आप ये बता देंगे कि फर्स्ट और सेकंड का वो मैं कैसे करूँ.
फर्स्ट और सेकेंड मेरा भी रिकॉर्डिंग वाला कुछ इशू हुआ था, तो आई हैव टू रिपीट दैट सेशन. तो यू कैन ज्वाइन in the later part of this class. I will let you know, because I've not recorded that session, so I have to repeat that class. So I will let you know. Okay, the same link will be applicable. Maybe the day will be changed. Okay, maybe I will take on Monday.
मैम एक्चुअली पता है मैंने इससे पहले भी पर इनरोल किया था, बट मेरा असाइनमेंट नहीं कर पाई थी. एक्चुअली मुझे इतना नॉलेज नहीं था कि असाइनमेंट ज़रूरी है, तो
Okay.
फिर मुझे सर्टिफिकेट नहीं मिल पाया था.
Okay.
तो फिर इस सेशन तो मैंने थोड़ा ध्यान दिया है, तो वही इसीलिए मैं पूछना चाह रही थी कि इस सेशन में जैसे थर्ड वाला जो थर्ड वीक चल रहा है उसका लास्ट डेट, मतलब असाइनमेंट का लास्ट डेट लिखा था. मैंने असाइनमेंट खैर कर ली है, तो
Okay.
उसका लास्ट डेट लिखा है, तो उससे पहले ही करना होता है ना.
हाँ जी.
और मैम ये रिकॉर्ड मतलब मेरा विजिबल रहेगा ना वहाँ पर, लाइक पोर्टल पे तो मेरे रहेगा ना.
हाँ, आई विल अपलोड, तो ये वाला रहेगा कि यू हैव अटेंडेड दिस सेशन.
ओके मैम. एंड लाइक अगर मैं लाइव सेशन अटेंड कर रही हूँ तो उसकी भी अटेंडेंस लगेगी ना.
यस.
ओके मैम, थैंक यू फॉर क्लियरिंग दैट. एक्चुअली पता है मुझे इस बात का आइडिया नहीं था.
ओके, नो इश्यूज.
ओके मैम, प्लीज यू कैन कंटिन्यू दैट.
So pick the odd one out from the given options: ring domain, HECT domain, U box, or destruction box.
क्वेश्चन क्या है मैम, फिर से रिपीट कर दें.
Pick the odd one out from the given options.
ओके, ऑप्शंस क्या है मैम.
The screen is not visible.
नो मैम, स्क्रीन इज नॉट विजिबल. एक्चुअली
Okay,
स्क्रीन बंद हो
wait.
गई है.
Is it visible now?
यस मैम.
Okay. So pick the odd one out from the given options.
मैम इसका आंसर मुझे नहीं पता.
Okay. It is destruction box because the ring domain, hack domain, and U box all types of these are E3 ubiquitin ligases, which are enzymes that transfer ubiquitin to a substrate protein, which is essential for degradation. While the destruction box, which is also known as D box, it is a sequence motif within the substrate protein, and it is recognized by the E3 ligase rather than it is a type of E3 ligase domain. So all these three are E3 ligase domain, while this is a sequence motif which is recognized by E3 ligase. Okay. So which of the following has been implicated in the regulation of histone stability for controlling gene expression?
In the regulation of these two मैम इसका क्वेश्चन जरा आप एक्सप्लेन करेंगे व्हाट डज इट मीन क्वेश्चन क्या कहना चाह रहा है
Which of the following has been implicated in the regulation of histone stability for controlling gene expression? Gene expression को कंट्रोल करने के लिए जो हिस्टोन स्टेबिलिटी रिक्वायर्ड होती है वो किस जीन के थ्रू होती है?
मैम हेट्रोटाइपिक ubiquinate chains.
It is all of the above. So let me explain. The regulation of histone stability for controlling the gene expression involves several mechanisms which is related to ubiquitin system only. So for the heterotypic and branched ubiquitin genes, these complex chains are where ubiquitin molecules are linked through different lysine residues or multiple sites, which are more efficient signals for degradation than homotypic genes. Okay, and talking about this K11 or 48, these are branched ubiquitin chains, and these have been emerged as a vital and enhanced degradation signal for target proteins, including histones, during cell cycle regulation. And this APCC, this is anaphase promoting complex or cyclosome. You might have heard of this. These are E3 ubiquitin ligase complex, and it is the specific enzyme which is responsible for assembling this K1148 chains on histones during mitosis. Okay, so these all regulate gene expression and maintain cell identity. So all of these components work together in this process. Okay. In which phase of the cell cycle does licensing of replication origin occur?
S phase.
This is late M or G1 phase. It is asking not about replication, it is licensing of replication. What does licensing of replication mean?
Okay, ma'am.
What does licensing means? It is the process that licenses DNA for replication, कि वो आगे proceed हो जाए. Not the DNA replication itself, not the DNA synthesis, it is the licensing. Okay, so it is the late MG1 phase where DNA replication begins once per cycle. During this phase, ORC जो origin recognition complex होता है, it binds to the replication origin and other proteins like CDT1, CDC6, and these help load the mini chromosome maintenance, which is MCM complex, and onto the DNA. So all these ORC, including CDT1, CDC6, and MCM complex, these bind to the DNA, and this complex is then activated at the start of the S phase to begin DNA synthesis, while the licensing it occurs in late M or G1 phase.
Yes, ma'am.
Coming to next, which of the following statements is incorrect for S phase of the cell cycle?
Yes, first, the phase also the synthesis phase red this separately regulated. Yeah. So once the cell phases go through the G1 restriction point, start to come to the removal of the external removal of the external stimuli, including entry into the S phase, cause the S phase cell back to the G1 phase.
Yes, right.
Once it means like अगर वो एक बार चला जाता है तो उसको वापस नहीं आता है ना, मतलब irreversible phase होता है ना वो तो.
So once a cell passes the G1 restriction point, start site, it is committed to complete the cell cycle, including the S phase, regardless of the removal of external stimuli like growth factors. So S phase is indeed known as the synthesis phase, and entry into it is highly regulated by checkpoints, which is cyclin CDK complexes. Okay, so all these are correct. S phase is also known as synthesis phase. Entry of a cell into the S phase is highly regulated. This is also correct. Once the cell passes through the G1 restriction point, it gets committed to go through the S phase. So fourth is the incorrect statement. Next, external growth factors induce the synthesis of D-type cyclin in mammalian cells. So this cyclin is important for
The cell growth, the cell growth restriction point, activation of ORC, and activation of two ligands. The external growth of the new the D cell, the close to ma'am, the cell to the cross the restriction point.
yes, correct.
Okay. External growth. Okay.
So D-type cyclins, we are talking about D-type cyclins which are synthesized in response to external growth factors. Okay, and then these bind to CDK4 or 6. So this whole complex is critical for pushing the cell cycle past the restriction point in the G1 phase. And at this restriction point, the cell commits to division regardless of the external factors. And this cyclin D, CDK4 or 6 complex phosphorylates the further retinoblastoma protein, which in turn inactivates the E2F transcription factor, and this allows the expression of genes necessary for S phase entry. So this whole complex, what this does, it phosphorylates the protein, and it in turn inactivates this E2F. Okay, so it is asking this cyclin is important for the cells to cross the restriction point. Further, the complex is important and not this particular cyclin. For the cyclin, it is important to push the cell cycle past the restriction point. Okay, so first is the correct option. Which is the catalytic subunit of 26S proteasome?
But 20 as subunit.
Yes, 20S subunit. So 26S proteasome, you might have heard of 26S proteasome. So this is composed of two main parts, 20S core particle and 19S regulatory particle. Okay, so one is the catalytic subunit, which is this 20S, and another is 19S, which is a regulatory particle. So 20S core particle is a hollow barrel shaped structure that contains the internal proteolytic chamber where protein degradation occurs. So it makes it this makes this 20S a catalytic subunit, while 19S regulatory particle, it is responsible for recognizing target proteins. and it feeds them into the 20th core for degradation. Okay.
Yes, ma'am.
Coming to next question, progression across G1 or S boundary followed by entry into S phase is promoted by activation of which of the following protein complexes?
Yes. Ma'am, CDK2 upon cyclic E.
Yes, correct. So you know the whole process?
यस मैम, एक्चुअली इसमें क्या होता है ना कि जो वो होती है एक्टिविटी होती है एसेंशियल की ना, वो ट्रांसलेशन होती है, शायद अभी इसमें पढ़ा था, रेप्लिकेशन ओरिजिन्स में वो क्या करती है, रेप्लिकेट होती है बेसिकली, ट्रांसफर होके एस फेज में जाती है, फिर वो जीवन और एस फेज का ट्रांजिशन हो जाता है.
Yes. So let me explain in detail. So the cell cycle, which is a highly regulated and ordered series of events, the engines that derive the progression from one step of cell cycle to next are known as cyclin CDK complexes. Okay, so these complexes are composed of two subunits. One is cyclin and another is CDPK, which is cyclin dependent protein kinases, which is known as CDK, cyclin and CDK complex. So cyclin is a regulatory protein, whereas CDK is a catalytic protein, and it acts as a serine threonine protein kinases. Cyclins are named as they undergo cycle of synthesis and degradation in each cycle. So humans contain four cyclins: G1, G1S, S and M cyclins. And explanation for this is cyclin CDK complexes trigger the transition from G1 to S phase and from G2 to M phase. So this question particularly asks about G1 or S transition. So D cyclins and CDK4 CDK regulate events in the early G1, and cyclin E CDK2 regulates the completion of the S phase. And transition from G2 to M, it is by the sequential activity of cyclin A and CDK1 and cyclin B CDK1. So this question might be modified in any of the uh, phases, so you should know everything that G1, if any question arises for the G1, you should know that cyclin D are responsible for this, cyclin D1, D2, D3, and CDK 4 and 6 are responsible. Okay, and if it asks about G1 to S transition, it is cyclin E and CDK2. If it is S phase, it is cyclin A and CDK2. If it is G2 to M transition, it is cyclin A and CDK1. And if it is M phase, it is cyclin B and CDK1. So it is memory based question, you just have to memorize all. Any of the question can come in your assignment. Okay.
यस मैम. मैम एक्चुअली वो बैक कर दीजिए, स्क्रीनशॉट ले
Yes,
लें.
yes.
एक सेकंड बोला तो एक सेकंड.
Is it fine?
एक्चुअली वो आपका वो पड़ा, वो स्क्रीन वो हो गई है. एक सेकंड. यस, एक मिनट बस.
Should I move forward?
एक सेकंड बस. सॉरी फॉर डिस्टर्बेंस. ओके मैम, थैंक यू सो मच.
Next is the same, the tabular form of the figure. So this is the figure which is this tabular form. It is the G1 phase, which denotes that cyclin D and CDK4 and 6 are responsible for this G1 phase. Okay, and moving to G1 to S phase, this is G1 to S phase transition. It is cyclin E and CDK2. And if I talk about S phase, it is cyclin A and CDK2. If I talk about G2 to M, it is cyclin A. G2 to M, it is cyclin B and CDK1. And M phase, it is cyclin B,
CDK1.
CDK1. So these are
Mhm.
spindle assembly checkpoints where chromosome attachment to spindle take place in presence of the cyclin and CDK complexes. So these particular checkpoints, there are three checkpoints here. Other than these transition, there are three checkpoints. One is it at G1 checkpoint, which is for the nutrients or DNA damage or growth factors. Another is at intra-S phase checkpoint, which is responsible for DNA damage or DNA replication checkpoint. Another is at G2, G2 to M transition checkpoint, and it is responsible for checking the cell size and DNA replication. Okay,
Yes.
so moving forward
So more.
from the this is from the week 2 assignment questions. Okay, so
मैम एक तो मैंने अटेंड नहीं किया, मतलब उसका सॉल्व भी नहीं किया मैंने अभी तक
okay,
प्रॉब्लम सेट। मैम
okay.
ये बताइए वीक 2 का असाइनमेंट क्या सॉल्व कर सकते हैं अभी भी?
इसका तो आपको मेल करके पूछना पड़ेगा, इसका आइडिया नहीं है मुझे। ओके ओके, सो week 2 I will be telling later then. Okay, in week 2 class. Now coming to the replication only. So you might have heard of DNA double helical structure, which is given by Watson and Crick model. So this helical structure, it has some key features, right? And this is a partial chemical structure where hydrogen bonded, where A is connected to D with 2 hydrogen bonds, while G is connected to C with 3 hydrogen bonds. Okay, and this is space filling model. So there are three different kind of structure of DNA double helical structure. So if I talk about the directionality of DNA from the 5 prime to 3 prime and 3 prime to 5 prime, it is 5 prime phosphate to 3 prime hydroxyl. So nucleotides in DNA backbone are bonded together by phosphodiester linkages between 3 prime and 5 prime carbons. And DNA molecule has direction. So complementary strands run in opposite directions. So talking about the DNA replication, it is the basis for inheritance. This is a fundamental process which occurs in all cells for copying DNA to transfer all the genetic information to daughter cells, and each cell must replicate its DNA before division. So DNA replication is semi-conservative in nature. Parental strands are not degraded. Base pairing allows each strand to serve as a template for a new strand, and new duplex is half parent template and half new DNA. You might know this thing. So I will in later slides I will tell in deep about the model from where this semi-conservative model came through. Okay, so DNA replication, it is semi-discontinuous. So in earlier slides it is semi-conservative, now it is semi-discontinuous. That is um, Okazaki fragments form in the when DNA replication starts. It is the leading strand, which is continuous synthesis, and in the opposite of this fragment, it is lagging strand, and Okazaki fragments form here. So further, these Okazaki fragments are joined by the ligases. So DNA replication is energy of replication. Nucleotides arrive as nucleoside triphosphate. DNA base sugar with triple P, triple phosphate, which is energy for bonding. And DNA bases arrive with their own energy source for bonding. It is bonded by enzyme DNA polymerase 3. So for DNA replication, primer is needed, DNA polymerase is needed, which can only add nucleotides to 3' end of a growing DNA strand. So it is important that DNA polymerase can only add nucleotides to 3' end. Now if you see this structure, DNA polymerase if add nucleotide to 3' end only, It is the leading strand where no disruption occurs, and it is known as continuous synthesis. While from here, if DNA polymerase start adding nucleotides from the 3' end, so it will form lagging strand, and it will be Okazaki fragments will form here, which will further be joined by the DNA ligases. Okay, so it needs a starter nucleotide to make a bond. The strand only grows 5' to 3', although it add nucleotides at 3' end, but it grows in 5' to 3' end direction. Okay, template is read in the 3' to 5' direction, while polymerization takes place in the 5' to 3' direction. So don't get confused with this direction. That template is read in 3' to 5' direction. Nucleotide is added at 3' direction from the 3' end, and polymerization occurs takes place in the 5' to 3' direction. Okay, clear this point? Is this clear?
Yes ma'am,
Okay.
एक्चुअली वो माइक बंद कर दिया था मैंने.
So now primer. So here RNA primer is required, which is synthesized by primase. It serves as a starter sequence for DNA polymerase 3. Only one RNA primer is required for the leading strand. RNA primers for the lagging strand depend on the number of Okazaki fragments. And RNA primer has a free 3' hydroxyl group to which the first nucleotide is bound. So these are the steps of DNA replication. First is the identification of origin of replication. Then unwinding, that is denaturation of double-stranded DNA to provide an ssDNA template. So first is this is DNA, double-stranded DNA. First is the identification of ori site, which is origin of replication. Okay, then it occurs that unwinding or denaturation of double-stranded DNA to provide a single-stranded DNA template. So this is single-stranded now. Formation of replication fork will happen. Then initiation of DNA synthesis and elongation. So if it is 3', DNA replication occurs like this. Then initiation of DNA synthesis and elongation. So it is DNA synthesis is happening and elongation further more will start like this and then this. Okay, then primer removal and ligation of the newly synthesized DNA fragments. So these DNA fragments will be joined with the help of ligases, and primer will be removed. Further reconstitution of chromatin structure. So this new strand, so this is new daughter template and this is new daughter template. So it will have half of the parent strand and half new DNA. It also has half of the parent strand and half of the new DNA. So reconstitution of chromatin structure then takes place after the DNA replication is completed. What are the components required for replication? First is the DNA polymerase. Without DNA polymerase, polymerization will not happen. Then helicase. Without helicase, unwinding will not happen. So if DNA, double-stranded DNA doesn't unwind, DNA polymerase cannot polymerize the fragment. Then topoisomerase. This relieves the torsional strain that results from helicase-induced unwinding. Then RNA primase. It initiates the synthesis of RNA primers. Then SS binding proteins, that is single-stranded binding proteins. It prevents the premature re-annealing of double-stranded DNA. If this happens, single-stranded binding proteins should be there. suppose if DNA polymerization starting from here and single stranded binding protein are not present, so this will reanneal. Okay, and then DNA ligase, which seals the single strand nick between nascent chain and the Okazaki fragments. So these Okazaki fragments will be sealed by the DNA ligase, and further the nascent chains. Now the origin of replication. If I talk about the prokaryotes, it is the origin of replication, which is ORI site. There is an association of sequence-specific double-stranded DNA binding proteins with a series of direct repeat DNA sequences. In E. coli, the ORI-C is bound by the protein DNA-A. So these are few important questions which can come. ORI-C. In E. coli, ORI-C is bound by the protein DNA-A. So this complex is formed consisting of 150 to 250 base pair of DNA and multiples of DNA binding protein. This leads to local denaturation and unwinding of an adjacent A plus T rich region of DNA. So it is talking about this complex or IC which is bound by DNA-A. So this complex is formed consisting of 150 to 250 base pair of DNA and multimers of DNA binding protein. So this leads to local denaturation and unwinding of an adjacent AT rich region of DNA. Now if I talk about origin of replication in eukaryotes, it is different site. So it is functionally similar autonomously replicating sequences which is known as ARS or replicators. In prokaryotes it is ORIC, in eukaryotes it is ARS or replicators, which have been identified in the yeast cells. So what does this ARS contains? It is autonomously replicating sequences. It contains a somewhat degenerate 11 base pair sequence which is known as origin replication element, ORE. Okay, then what does ORE do? ORE binds a set of proteins which is analogous to DNA-A. DNA-A, so this is kind of ORIC which is in prokaryote, and DNA-A, it is ORE binds to a set of proteins which is analogous to DNA-A, protein of E. coli. It is known as ORC. So if we distinguish origin of replication between prokaryotes and eukaryotes, origin of replication, ORIC. Here it is ORIC, here it is ORE. Here it is DNA-A, and here it is ORC. Okay, so this ORE is located adjacent to an approximately 80 base pair 80 rich region that is easy to unwind, and this is known as DNA unwinding element. DNA unwinding element. Okay, so this is the difference between prokaryote and eukaryote. Further, unwinding of DNA. The interaction of proteins with ORI defines the start site of replication and provides a short region of ssDNA. And this ssDNA further will require the single-stranded DNA binding proteins which stabilize this complex, where this single-stranded DNA binding proteins will bind to the single strand so that it doesn't wind again. Okay, so the interaction of proteins with ori defines the start site of replication and provides a short region of single stranded DNA, which is essential for initiation of synthesis of the nascent DNA strand. Then DNA helicase allows for processive unwinding of DNA. DNA helicase, you know that it unwinds the DNA. For the single stranded DNA binding protein, it stabilizes complex, and incorporation with this single stranded binding proteins, this leads to DNA unbinding and active replication. So all this complex, this leads to DNA binding and active replication starts up. So unbinding of DNA, replication fork will take place. DNA helicase, it unwinds the DNA and replication fork forms. So formation of the replication fork, the polymerase 3 holoenzyme, it binds to template DNA as a part of multi-protein complex. This DNA polymerase is only synthesized DNA in the 5' to 3' direction because the DNA strands are antiparallel. The polymerization functions asymmetrically. On the leading strand, the DNA is synthesized continuously. It is 3 prime, it is 5 prime. So this is the leading strand. Here DNA is synthesized continuously. On the lagging strand, the retrograde strand, the DNA is synthesized in short fragments. So this is known as Okazaki fragments. So this is the clear figure which shows all the components required for the replication and further their steps. First of all, helicase is unwinding the parental double helix. Okay, so this is helicases which unwinds the parental strand. So it starts from here and it starts to unwind the parental double strand. Then this is the single strand protein which stabilizes the unbound parental DNA. Then third, the leading strand. The leading strand is synthesized continuously in 5' to 3' direction by DNA polymerases. So we need helicases, we need single stranded binding proteins, DNA binding proteins. Third, we need DNA polymerases. Then the lagging strand is synthesized discontinuously. So here we require primase which synthesize a short RNA primer which is extended by DNA polymerase to form an Okazaki fragment. Now when Okazaki fragment is formed, we require DNA ligase to join these Okazaki fragments. Now after the RNA primer is replaced by DNA ligases join the Okazaki fragments to grow strand. Overall direction of replication is this 5' to 3'. So components we require, one is helicase, second is, you speak, which components we require for DNA replication,
Yes, ma'am.
which components are required for DNA replication.
Hmm. DNA ligase.
Ligase, yes.
और प्रोटीन हेलिकेस
Helicase,
लाइकेस
ligase,
प्राइमेज
primase. What does primase do?
जी
What does primase do?
मैम व्हाट डज द प्राइमेज डू
Yes.
मैम इट इज सिंथेसाइज और रेप्लिकेट
You're talking about primase or polymerase?
मैम प्राइमेज
Primase. What does primase do?
मैम इट रेप्लिकेट
Primase synthesizes a short RNA primer, which is required here for for the Okazaki fragment. So this primase synthesizes a short RNA primer which is extended by DNA polymerases to form an Okazaki fragment.
ओके
Okay. Another component,
मैम प्रोटीन
protein.
नहीं नहीं सॉरी सॉरी हेलिकेस हो गया लाइकेस हो गया अब प्राइमेज हो गया मैम पॉलीमरेज
Polymerase, right. Polymerase. Another component,
जब नहीं पता
everything is in this slide only. Everything is in front of you.
मैम आरएनए प्राइमर
Primer is synthesized by primase, so primase is important. Helicase is important. Second is SS binding proteins, single stranded binding proteins. If these are not present, the helicase which unbinds the parental double strand, it will bind again. So these are important, which maintains the single stranded binding, which maintain the single stranded DNA in its form only. Okay, so that parental strand doesn't bind again. Okay, so helicase is required, then single stranded binding proteins is required, then DNA polymerase, primase, and then DNA ligase. Okay, clear?
यस
So it is SS binding proteins.
एस एस बाइंडिंग प्रोटीन ओके मैम
Okay, so formation of replication bubble. When replication occurs, it occurs in bond direction along the length of DNA, and both strands are replicated simultaneously. So this replication process generates a replication bubble, which you have seen here. This is the replication bubble. Okay, so this is the origin of replication, and this is the bubble which is formed with the help of helicase. Now this is the parental strand, this is the daughter strand. So replication fork is this. Fork is where half part is uh, winded and half is unwinded. So this is replication fork, and replication bubble is this part. So here two daughter DNA molecules are formed. In eukaryotes, DNA replication begins at many sites. So this is ORI site. Here it is also origin of replication. Here it is also. So at many places DNA replication occurs along the giant DNA molecule of each chromosome. While in prokaryotes, it is at single site, which is ORIC. Further, DNA polymerase complex. There are number of different DNA polymerase molecules which engage in DNA replication. So these share three important properties: chain elongation, processivity, and proofreading. So all these three properties are present with the DNA polymerase, which are involved in the DNA replication. One is chain elongation. So chain elongation accounts for the rate at which polymerization occurs, which means that how many nucleotides per second are added at each chain. So this is the chain elongation. that is the rate of polymerization. Okay, second is processivity, which is an expression of the number of nucleotides added to the nascent chain before the polymerase disengages from the template. That is, processivity is number of whole nucleotides which are added to a new chain. Suppose this much polymerase
Ma'am, I
has.
will be back in just a 5 minutes, please.
Okay.
Yes, ma'am. Thank you for understanding. Hello. Hello. Ma'am, am I audible? Sorry, ma'am. Am I audible? Hello. Are you there, ma'am? Hello. Am I audible, ma'am?
Can we continue?
Hello.
Okay.
Hello. Hello.
Yes. Am I audible?
Ma'am.
Yes.
Hello. हेलो।
Yes.
एक्चुअली मैम आप विजिबल नहीं हो रही थी, मैंने 3 बार रीज्वाइन किया फिर आप आई।
Okay. Is the screen visible?
Yes, ma'am.
Okay. So we were talking about the DNA polymerase and its important properties. So we
Yes, ma'am.
started with the chain elongation, which is the rate at which polymerization occurs. That is nucleotides which are added per second. Then processivity, which is an expression of the number of nucleotides added to the nascent chain before the polymerase disengages from the template. So this is the processivity, that uh, number of nucleotides added to the new chain, right, before the DNA polymerase get removed from the chain. So this um, DNA polymerase is removed from the chain and the number of nucleotide it added to the new chain, right? So it is the processivity of that particular polymerase. Chain elongation is the rate at which polymerization occurs, that is per second the number of nucleotides added. Processivity is the number of nucleotides which are added before it get disintegrate from the template. And proofreading, it is a function which identifies the copying errors and it corrects them. So there are three important properties which DNA polymerase has. One is chain elongation, it is the rate at which nucleotide are added per second. Second is processivity, which is number of nucleotides added before the polymerase is disintegrated. And third is the proofreading, which it identifies the copying errors and corrects them before it gets disintegrated. Next is the DNA polymerase complex. In E. coli, polymerase 3, it functions at the replication fork. So, of all the polymerases, it catalyzes the highest rate of chain elongation and it is the most processive. In E. coli, polymerase 3, it functions at the replication fork. So, when it is a replication fork and DNA polymerase 3 is there, so of all the polymerases, it catalyzes the highest rate of chain elongation and is the most processive. So till now we have studied about three important properties of DNA polymerase. One is chain elongation, another is processivity, and third is proofreading. Okay, so in E. coli, polymerase 3 has the highest rate of chain elongation and is the most processive. Now, polymerase 3 and polymerase 2, it is the mostly involved in proofreading and DNA repair. And polymerase 1, it completes chain synthesis between Okazaki fragments on the lagging strand. So polymerase 1, it completes the chain synthesis between Okazaki fragments on the lagging strand. DNA polymerase 2, it is involved in the proofreading and DNA repair. And polymerase 3, which is mainly involved in the chain elongation and is the most processive in E. coli. Okay, this point is clear?
Yes, ma'am.
Okay. So now difference between DNA polymerase 1, 2 and 3. So polymerization rate. So as we already studied that polymerase 3 is uh, has the high chain elongation rate and then is highly processive. So DNA polymerase 3 is high in these while both of these is low. Then talking about the proofreading is present in all. All of these polymerases have 3' to 5' exonuclease activity, which is important for proofreading, while DNA polymerase 2 is mostly involved in proofreading. Mostly this is involved while all has this property, and only polymerase 1 has 5' to 3' exonuclease activity, while all has 3' to 5'. Now primer removal, which is best done by DNA polymerase 1. Then strand synthesis. DNA polymerase 3 can synthesize strand in both the directions, while polymerase 1 can only do in the lagging strand. And DNA polymerase 2 has no role in synthesis. Now DNA repair. So DNA repair, polymerase 1 and 2 are mainly involved and not 3. So this is very important. Now eukaryotic DNA polymerases. Eukaryotic cells have counterparts for each of these enzymes plus some of the additional ones. So E. coli DNA polymerase 1, 2 and 3. Now 1. Now in mammalian, 1 is alpha, which is involved in gap filling and synthesis of lagging strand. 2 is epsilon. 2 is epsilon, beta and gamma in eukaryotes. So epsilon is involved in proofreading and repair, beta is involved in DNA repair, gamma is involved in mitochondrial DNA synthesis. And DNA polymerase 3, which is DNA polymerase 3 in E. coli, while delta in mammalian. So it is highly processive and leading strand synthesis, As we know that DNA polymerase 3 is highly processive and has high chain elongation in E. coli, so here delta is mainly involved in eukaryotes. So initiation elongation of DNA synthesis. So if I talk about primer, the priming process involves the nucleophilic attack by 3' hydroxyl group of the RNA primer on the phosphate of the first entering deoxynucleoside triphosphate with the splitting of pyrophosphate. So the priming process which involves the nucleophilic attack. If you remember, I have shown here. I will continue in 5 minutes.
Okay, ma'am, when the class will be over?
Around 8. Around 9, sorry.
Okay.
Can I start?
Yes, ma'am.
Okay. So my screen is visible?
Yes, ma'am.
Okay. So I was talking about the phosphate group and hydroxyl group. So it is the nucleophilic attack. So it is the priming process. It is the nucleophilic attack by the 3' hydroxyl group of the RNA primer on the phosphate group of the first entering deoxynucleoside triphosphate. So this is one nucleoside, this is another nucleoside. So this has hydroxyl group. Now this here it has hydroxyl group and here it has phosphate group. So it is a nucleophilic attack of the hydroxyl group on the uh, with the phosphate group of entering nucleoside, right? So both of this hydroxyl group of one nucleoside and phosphate group of another nucleoside combine together. So here it is. It is the nucleophilic attack by the 3' hydroxyl group of the RNA primer on the phosphate of the first entering deoxynucleoside triphosphate with the splitting off of pyrophosphate. Then mammalian DNA polymerase alpha. Now it is talking about alpha. Alpha which is involved in gap filling and synthesis of lagging strand. So this DNA polymerase alpha, it is mainly responsible for the synthesis of primer. So then it enters. It is in the process of initiation and elongation of DNA synthesis. Then selection of the proper deoxyribonucleotide whose terminal 3 hydroxyl group is to be attacked is dependent upon proper base pairing with the other strand of the DNA molecule according to the rules proposed originally by Watson and Crick. So it is saying that if A is there, it should bind with T, and if G is there, it should bind with C. So when an adenine deoxyribonucleoside So when an adenine deoxyribonucleoside monophosphoryl moiety is in the template position, a thymidine triphosphate will enter and its phosphate will be attacked by the 3' hydroxyl group. Can you mute please?
Mute is mute.
Okay. So by this stepwise process, the template dictates which deoxyribonucleoside triphosphate is complementary, and by hydrogen bonding, which which another triphosphate is required. Like if adenine deoxyribonucleoside monophospholate moiety is there in the template position, so thymidine triphosphate will enter and its phosphate will be attacked by the 3' hydroxyl group. Okay. So this is the base pairing in DNA replicant, like this. So if this is the double stranded DNA, helicase will unbind this, then nucleotide will be added like this. Phosphate will be attacked with the hydroxyl of this, and this is how it enters. Now talking about DNA topoisomerase. It is the relief of supercoil which is done by topoisomerase. So there are two type of topoisomerase, 1 and 2. Topoisomerase 1, which acts by making a transient single cut in the backbone of the DNA, enabling the strand to swivel around each other to remove the buildup of twist. While topoisomerase 2, which is known as DNA gyrase, it acts by introducing double-stranded breaks, enabling one double-stranded DNA to pass through another, thereby removing knots and entanglements that can form within and between DNA molecules. So it is basically involved in the relief of supercoils. So two types of topoisomerase, 1 and 2. One is which is involved by making a single cut in the backbone of the DNA, and it enables the strand to swivel around each other to remove the buildup of twist. While topoisomerase 2, which is also known as DNA gyrase, it acts by introducing double-stranded break, enabling one double-stranded DNA to pass through another and thereby removing knots and entanglements that can form within and between DNA molecules. So it involves single stranded cut and it involves double stranded cut. So formation of supercoil. This is DNA double helix. This is how supercoil forms. This is strand separation, and if strand separated, it will be positive supercoiling will happen here. So topoisomerase will be needed. So topoisomerase 1, if 1 is present, it will create single stranded cut, and this nick, what this nick will do, first the left half of the circle folds over the right half, then the back half of the helix is cleaved, and third, the front half of the helix passes through the break, which is resealed again, like this. So it is like this first, and then after cut it will be negatively supercoiled now, then the neck will be sealed. So now the primer removal and nick sealing. The primers are removed by DNA polymerase 1, which we have already studied while talking about different kinds of DNA polymerase and its function. So primers are removed by DNA polymerase 1 by replacing ribonucleotides with deoxyribonucleotides, and nicks are sealed by DNA ligase, as you know, and then multiple primers on the lagging strand while single primer on the leading strand. Now proofreading and editing. 1000 bases per second. DNA polymerase 1. It proofreads and corrects the typos. It repairs mismatched bases. It removes abnormal bases. Repairs damage throughout life. It reduces error rate from 1 in 10,000 to 1 in 100 million bases. Now termination of replication. In prokaryotes, DNA replication terminates when replication forks reach specific termination sites. Like there is an origin of replication, there is a termination site also, where the replication terminates when replication fork reach some specific termination sites. Now the two replication forks meet each other on the opposite end of the parental circular DNA. This process is completed in about 30 minutes. A replication rate is 3 into so the entire mammalian genome replicates in approximately 9 hours. The average period required for formation of a tetraploid genome from a diploid in a replicating cell. Now reconstitution of chromatin structure is the last step of replication, which is chromatin structure must be reformed after replication. Now there is newly replicated DNA which rapidly assembled into nucleosomes, and the pre-existing and newly assembled histone octamers are randomly distributed to each arm of the replication fork. So now it is saying that newly replicated DNA. Now we have newly replicated DNA like this. It is rapidly assembled into a nucleosome, and the pre-existing and the newly assembled histone octamers are randomly distributed to each arm of the replication fork, like this. Now DNA synthesis and the cell cycle, which we started with, that DNA synthesis happens in the S phase. Now if we talk about the animal cells including human cells, the replication of the DNA genome occurs only during the S phase, which is a synthetic phase. This is usually temporarily separated from the mitotic phase by non-synthetic periods like G1 and G2, occurring before and after the S phase. The cell prepares for DNA synthesis in G1 and for mitosis in G2. So the cell prepares for synthesis in G1 and for mitosis in G2. Now telomeres. In eukaryotic replication, where the removal of RNA primer from 5' end of lagging strand, a gap is left. This gap exposes the DNA strand to attack the 5' exonucleases. This problem is overcome by the telomerases. We have already talked about the telomerases. Now, this telomerases, the gap which is present when the last nucleotide is added, which exposes the DNA strand to attack the 5' exonuclease. So here it is the DNA. Now telomeres are there and newly synthesized strand with terminal RNA primer is removed. So repeating non-coding sequences at the end of chromosomes, which is a protective cap, it limit to around 50 cell divisions. Mechanism of action of telomerase. So now telomerase comes in action. RNA template. It is an RNA template part of telomerase. The enzyme synthesizes this TT, TTA, triple G into N. This repeat and onto the telomere sequences using an internal RNA template. Now telomerase, now again this is added, then second telomere is added, then third telomere is added. Telomerase acts like a reverse transcriptase. It recognizes 3' end of the telomere based on the RNA component. A small DNA strand is synthesized here. So it should stop somewhere. Now telomerase should not be highly active that it keeps on adding the telomeres. So different telomerase have different level of activity in different cells. So it is high in stem cells and cancer cells, and activity is lost at old age and potential target for newer anti-cancer drugs. Now coming to the inhibitors of DNA replication. So as you know that type 2 topoisomerase, which is known as DNA gyrase, it is inhibited by novo, biosin and nalidixic acid. So this is a memory based, it can come in your assignment question. So DNA gyrase, which is a type 2 topoisomerase, it is inhibited by novo, biosin or nalidixic acid. Then ciprofloxacin, it interferes with the DNA breakage and rejoining processes. Then talking about mammalian topoisomerases, which is inhibited by etoposide and adriamycin, which is used as an anti-cancer drugs. Nucleoside analogs also inhibit replication and are used as an anti-cancer drugs. So these are the inhibitors of DNA replication. So now come summarizing the replication whole. The unwinding forms the replication fork. Primase synthesizes the RNA primer. Continuous synthesis will happen on the leading strand. Discontinuous synthesis will happen on the lagging strand, and it forms the Okazaki fragments. Then synthesis, DNA polymerizes or synthesizes in 5' to 3' direction. Primers need to be removed, nick need to be sealed. Proofreading by DNA polymerases, different kind of DNA polymerases are required for proofreading. Then it is organized into a chromatin structure. So this is the summary of replication. Now if I talk about replication facts, now summarizing whole in few different facts. So DNA has to be copied before a cell divides. DNA is copied during the S or synthesis phase of the interphase. New cells will need identical DNA strands. Now there are different models of replication, as we have already in the initial slides I have told that it is semi-conservative. So there were three different models of replication, which was semi-conservative, conservative, and dispersive model. So in the semi-conservative mode of replication, in this model, two strands of DNA unwind from each other, and each acts as a template for synthesis of a new or complementary strand. This results in two DNA molecules with one original and one new strand, which we use to study, and we call that DNA semi-conservative in nature, that it follows the semi-conservative replication. Now talking about the conservative replication, it in this model, DNA replication results in one molecule that consists of both original DNA fragments identical to original DNA, and another molecule that consists of two new strands with exactly the same sequences as the original molecule. So it is conservative. Like we study that one is parental and one is original. One is parental and one is newly synthesized. While in conservative, it says that DNA synthesized like this. So this will be one, and this another will be one. Parental different and newly synthesized will be different. So it is conservative replication. Now talking about dispersive replication, in the dispersive model, DNA replication results in two DNA molecules that are mixture or hybrids of parental and daughter DNA. In this model, each individual strand is a patchwork of original and new, like this. So this is original, this is new. Suppose this green one is new and red is original. So dispersive is this kind. While semi-conservative is one parental, one new. Conservative is parental different, original, parental different, new different. Semi-conservative, one parental, one new. And dispersive is this, patches of new, patches of original and new. So it is kind of hybrid. So from all these models, then DNA replication is finally said to be semi-conservative, meaning that each strand in the DNA double helix acts as a template for the synthesis of a new or complementary strand. Conservative is this, semi-conservative is this, and dispersive is this. So DNA is semi-conservative. DNA replication is semi-conservative in nature. So this process takes us from one starting molecule to two daughter molecules, with each newly formed double helix containing one new and one old. So this is one old and one new, one old and one new. So this is the Michelson-Stahl experiment, which proved that DNA replicates semi-conservatively. They grow bacteria in heavy nitrogen medium, and then transfer some bacteria to light nitrogen medium. Bacterial growth continues. Then take samples 0 minute, 20 minute, 40 minutes, like one round of replication, two round of replication. And then before the bacteria reproduce for the first time in the light media, all DNA is heavy. So after two generations, half the DNA was intermediate and half was light. So there were no heavy DNA. Like this was parental strand, and new strand was the light nitrogen medium. So this experiment proved that DNA replicates semi-conservatively and not conservatively. So this pattern could only have been observed if each DNA molecule contains a template strand from the parental DNA, and thus proved that DNA replication is semi-conservative in nature. So this is DNA double helix, hydrogen bond breaks and DNA helix opens. Each strand of DNA acts as a template for synthesis of a new complementary strand. So polymerization occurs in 3' to 5' direction, like it is in 5' to 3 direction, but nucleotides are added at 3' direction. So replication produces two identical DNA double helixes, each one with one new and one old strand. So this DNA polymerases, I have already talked about it. They always need a template. They can only add nucleotide to 5'. They can't start making a DNA chain from scratch, but require a pre-existing chain or short stretch of nucleotides called a primer. And they also have a proofreading function also. So they check their work, removing the vast majority of wrong nucleotides that are accidentally added to the chain. So we have already studied this prokaryotic and eukaryotic DNA polymerases 1, 2 and 3. So it is gap filling following DNA replication, repair and recombination. 2 is DNA proofreading and repair, and here it is beta and gamma. So it is DNA repair and mitochondrial DNA synthesis, while the one which is polymerase 3 for E. coli, it is epsilon for eukaryotic, which is processive and it synthesizes the leading strand. The DNA G, which is primase, plays the role of primase in E. coli. It is alpha in eukaryotes, and delta, which is processive and lagging strand synthesis happens with this. So you should remember all these: alpha, beta, gamma, epsilon and delta, and all 1, 2, 3 and their functions. So this is very important. So this I already told. This is replication form. Replication bubble form. Prokaryotes have a single bubble, eukaryotes have many bubbles, because eukaryotes have many orysites, origin of replication, while prokaryotes have single origin of replication, and this single bubble will form, while eukaryotes have different origin of replication and different bubbles will form. Enzyme helicase, which unbinds by breaking the weak hydrogen bonds. Then single-stranded binding proteins attach and keep the two DNA strands separated and untwisted. Then primers and primases, this I have already told. Leading and lagging strand is already covered. So this is the leading, this is lagging Okazaki fragments. So other than these proteins, the sliding clamp which holds the DNA, this term didn't come till now. The sliding clamp which holds the DNA polymerase 3 molecules in place as the synthesized DNA. This sliding clamp is a ring-shaped protein and keeps the DNA polymerase of the lagging strand from floating off when it restarts at a new Okazaki fragment. So this is also important. Now topoisomerase, it is already known that during DNA replication it plays a role of important maintenance. This enzyme prevents the DNA double helix ahead of replication fork from getting too tightly wound as the DNA is opened up. So it makes the temporary nicks, either 1 or 2 depending on the type of topoisomerase it is. That one, the topoisomerase 1 creates single-stranded, creates nick on the single-stranded, and topoisomerase 2, which is DNA gyrase, it breaks the double strand to release the tension and then seals the nick to avoid permanent damage. Then RNA primers are removed and replaced by DNA through the activity of DNA polymerase 1. The nicks that remain after the primers are replaced get sealed by the DNA ligase. Now helicase, this is the function. These are all the components which I have already discussed. So you should know that helicase opens up the DNA. Single-stranded binding proteins are important, so that now you should tell what helicase does one by one. What is the role of helicase? What is the role of helicase? It unwinds the DNA. Then single stranded binding protein, it it is present to prevent rewinding of the DNA. Topoisomerase works at the region ahead of the replication fork. It prevents a supercoiling. Primase synthesizes RNA primers, which is complementary to the DNA strand. DNA polymerase 3 is extend the primers, adding on the 3' end to make the bulk of the new DNA. And RNA primers are removed and replaced with DNA by DNA polymerase 1. Then gaps are filled and sealed by DNA ligases. Then DNA proofreading and repair. So why this proofreading and repair is very important? As replication occur, errors and DNA damage are actually happening in the cells of our body all the time. In most of the cases, they don't cause cancer or even mutations. That's because they are usually detected and fixed by DNA proofreading and repair mechanisms. While if the damage cannot be fixed, the cell will undergo PCD, which is programmed cell death, that is apoptosis, to avoid passing on the faulty DNA. Now mutations happen and get passed on to daughter cells only when these mechanisms fail. Cancer, in turn, develops only when multiple mutations and division related genes accumulate in the same cell. Now, proofreading. During DNA replication, most DNA polymerases can check their work with each base that they add. This process is called proofreading. So, polymerase add an incorrect nucleotide to the new strand of DNA. Then, polymerase detect that bases are mismatched, and then polymerases uses 3' to 5' exonuclease activity to remove incorrect nucleotide. Before continuing with the DNA synthesis, it removes that incorrect nucleotide. Even if after that this nucleotide is not removed, then another polymerase will come and play the role of 3' to 5' exonuclease activity and plays the role of proofreading, and then this incorrect nucleotide will be removed. So, mismatch repair. This happens right after new DNA has been made, and its job is to remove and replace mispaired bases, ones that were not fixed during proofreading. Mismatch repair can also detect and correct small insertions and deletions that happen when the polymerase is losing its footing on the template. In bacteria, original and newly made strands of DNA can be told apart by a feature called methylation state. An old DNA strand will have methyl groups attached to some of its bases, while a newly made DNA strand will not have yet gotten its methyl group. So, in eukaryotes, the processes that allow the original strand to be identified in mismatch repair, it involves a recognition of nicks, which is single stranded breaks, and these are found only in the newly synthesized DNA and not in the old one. So, if a mismatch is detected in newly synthesized DNA, the new DNA strand is cut like this, and the mispaired nucleotide and its neighbor are removed. The missing patch is replaced with correct nucleotides by another DNA polymerases, like three polymerases have different role. So proofreading one will replace this, and the gap will be sealed with the DNA ligase. Now, DNA damage repair mechanism. Earlier it was uh, mismatch repair. Now it is DNA damage repair. So some DNA damaging chemical reactions can be directly undone by enzymes in the cell. Damage to one or a few bases of DNA is often fixed by removal or excision and replacement of the damaged region. While in base excision repair, just the damaged base is removed. While in nucleotide excision repair, as in the mismatch repair we saw above, a patch of nucleotides is removed. So double stranded break repair two major pathways are there: non-homologous end joining and homologous recombination, which are used to repair these double stranded breaks in DNA. And that is when an entire chromosome splits into two pieces. So there are different kind of damage repair mechanism. So reversal of damage: DNA damage often just involves an extra group of atoms getting attached to DNA through a chemical reaction. For example, guanine can undergo a reaction that attaches a methyl group to an oxygen atom of the base. So when there is this methyl bearing guanine, if not fixed, it will pair with the thymine rather than the cytosine. Luckily, humans and many other organisms have an enzyme that can remove the methyl group, reversing the reaction and returning the base to normal. So, this only methyl group can lead to mismatch, because methylated guanine cannot bind to cytosine, rather, it will bind to thymine. Now, base excision repair. It is a mechanism used to detect and remove certain types of damaged bases. A group of enzymes called glycosylases. This is a this play a key role in base excision repair. Each glycosylase detects and removes a specific kind of damaged base. For example, chemical reaction called deamination can convert a cytosine base into uracil, a base typically found only in RNA. During DNA replication, uracil will pair with adenine rather than guanine. So, an uncorrected cytosine to uracil change can lead to mutation. So, to prevent such mutations, glycosylase is there, which is a group of enzyme, and from the base excision repair pathway, it detects and removes deaminated cytosines. Once the base has been removed, the empty piece of DNA backbone is also removed, and this gap is filled and sealed by other enzymes. So here deamination converts a cytosine base into a uracil. So now uracil is bind to guanine. Now uracil is detected and removed, leaving a baseless nucleotide. The baseless nucleotide is removed, leaving a small hole in the DNA backbone. Now the hole is filled with the right base by a DNA polymerase, and the gap is sealed by the DNA ligase. Now nucleotide excision repair. This is something which is used to fix some type of damage which is caused by UV radiation. Suppose when you get a sunburn, UV radiation can make cytosine and thymine bases react with neighboring bases that are also C or T, forming bonds that distort the double helix and cause errors in DNA replication. The most common type of linkage which happens is thymine dimer, consists of two thymine bases. So in nucleotide excision repair, the damaged nucleotides are removed along with the surrounding patch of DNA. In this process, a helicase cracks the open to form a bubble, and DNA cutting enzymes chop out the damaged part of the bubble. So further, DNA polymerase replaces this missing DNA, and DNA ligase seals the gap in the backbone of the strand, which reacts with each other, and then this become chemically linked. Here, as it is, UV radiation produces a thymine dimer, and once the dimer has been detected, the surrounding DNA is open to form a bubble, and then enzymes cut the damaged region out of the bubble. Now polymerase will replaces the excised cut, and ligase seals the backbone. Now double stranded break repair. It is another
मैम कितनी देर में लेक्चर खत्म होगा? एक्चुअली 8:50 में.
10 minutes more.
ओके, उससे पहले नहीं हो सकता?
Okay, I will try to wrap up. So
Okay,
this is the
please.
last double stranded break repair, which is present in CRISPR also. So these double stranded breaks are dangerous because large segments of chromosomes and hundreds of genes they contain may be lost if break is not repaired. So there are two pathway for repair mechanism of double stranded break. One is non-homologous end joining, and other is homologous recombination. So this non-homologous end joining, the two broken ends of chromosome are simply glued back together. This repair mechanism is messy and typically involves the loss and sometimes addition of a few nucleotides at the cut site. So non-homologous end joining tends to produce a mutation which could be deletion, insertion or replacement. So this is better than the other alternative, loss of an entire chromosome arm. While in homologous recombination, there is a two homologous chromosomes come together and undamaged region of the homolog or chromatid is used as a template to replace the damaged region of the broken chromosome. So homologous recombination is cleaner than non-homologous end joining and does not usually cause mutations. So we have studied about base excision, mismatch repair and nucleotide excision. So all these are the repair mechanisms. We have already talked about telomerases. So this is the whole process which we have talked about: helicase, then single-stranded break, then DNA polymerases, then primase, and then DNA ligase. This is the kind of mutation which can happen, like alkylating agent which can add methyl group and can lead to methylguanine which can bind to which cannot bind to cytosine. Then X-rays and other rays calling dimer of thymine, then AG mismatch, TC mismatch. So all these will be repaired by different repair mechanisms. So this is the last slide where I want to um, talk about the Nobel Prize which was in chemistry in 2015, which was awarded to this group for having the mapped how cells repaired damaged DNA. So DNA damage and then base excision repair, nucleotide excision repair and mismatch repair. So this group won the Nobel Prize in chemistry in 2015 for this discovery. So thank you for today's lecture and thank you for attending.
Thank you.
Okay, so for the other week 1 and week 2, I can you can share your email ID in the chat and I will let you know through the same link you can join. And for week 1 or 2, if you want to join, you can let me know. You can enter your email ID. Okay.
मैम वो कह रहे थे कि वो असाइनमेंट्स सारी वो क्या करना चाह रहे थे, हाँ असाइनमेंट्स कितनी होनी चाहिए टोटल?
टोटल 5 हैं जो आपके पोर्टल पे अवेलेबल हैं.
थैंक यू मैम.